Service correspondent Jewel Fraser, and in this IPS podcast, I talk with a scientist from the International Livestock Research Institute in Nairobi, Kenya. Dr. Jean-Baptiste Entfellner and his colleagues are doing work they hope will encourage Africans to make greater use of the continent's indigenous crops. They hope Africa can be helped to forego the imported foods that are popular globally and rely more heavily on foods native to the continent, thus boosting food self-sufficiency. Nearly 30% of the world's population experience food insecurity, with Africa's population being nearly twice as likely as the global average to do so, says a recent FAO report. Could turning to local crop varieties be a solution? Also, let me give a shout out to my colleague Crystal Onkyo, a Kenyan journalist who helped me to get this interview recording done. All right, let me know when you're ready, Crystal, when I can go.
I'm ready.
Okay, I am Jean-Baptiste Entfellner, a bioinformatics scientist by training, and currently here at the International Livestock Research Institute, I am assuming the role of head of the data and research methods.
Thank you. Now, can you share with us the background to the genomic sequencing work being done on crops at your institute and why this work is considered necessary?
So at the International Livestock Research Institute, we not only research livestock, but also feed and forages. So these sources of food that will be key to the livestock, particularly in the African continent. And in this context, we have been working on lablab. So the lablab is a species of bean that is very much present in all of Africa, but also in South Asia, especially in India. That crop is a very interesting one because it's used, of course, by farmers to feed their families thanks to the beans themselves. But also that crop has a very high protein content even after you have removed the beans. So the remaining parts of the plant, the stem, leaves, etc., can be given as forage to our livestock, especially cattle and goats also. So it was a very interesting project we led, and that unveiled the whole genome sequence of lablab. For the first time, our team, composed of a majority of African researchers and led by African researchers, our team was able to decipher the whole genome of that crop. The genome is like a map for scientists to try and find their ways in the biological information that characterizes a species, in this case, the lablab bean.
I'd like you to speak more broadly as to why the Institute considers it's important to do this genomic sequencing across the different types of food available.
So genomics is about reading the genome information for any species, be it for livestock or for humans or for plants and fish, etc. In my institute, because we are the International Livestock Research Institute, we are most concerned with species of agricultural interest. And you may know that the genome is this very long series of A, C, G, and T letters. And the human genome comprises more than 3 billion of these letters. They are organized in such a way that they encode for genes, and those genes in turn produce proteins. Now, in any organism, those genes are at the basis of all the biological pathways. Everything that takes place, let's say, in an organism is directed by the genome. The genome is like the recipe or the cookbook, actually, to cook so many recipes that are crucial for our organisms to function. So there are genes responsible for susceptibility to some diseases. And in the case of lablab, what was interesting for us is that there are genes responsible for the synthesis of compounds that are anti-nutritional. For instance, chemical compounds that will be difficult to digest, or chemical compounds that make it that you'll need a very long cooking time for a certain crop. So it's very important to know where these genes are on the genome in order to inform, for instance, breeders who will try to make crosses to improve the agricultural value of a certain species.
Okay, so which organizations provide support for the Institute's work?
Well, we have a number of funders. Some projects are funded bilaterally. Some projects are funded by funds gathered by the CGIAR consortium. We're part of a consortium of institutes, initially called a Consultative Group for International Agricultural Research. And we have funders such as BMGF, the Bill and Melinda Gates Foundation. We have the World Bank in some projects. We have USAID. We have the German cooperation, the Swiss cooperation, the Swedish cooperation, a number of them. And inevitably, when I start listing them, I forget many.
I can imagine. All right. So now, the Nature article mentioned the focus on involving African researchers in the LabLab project. How was that achieved?
Great. So, we firmly believe at the International Livestock Research Institute that one of our missions is to enhance the capacity to do research by African scientists. So this project on the LabLab sequencing and annotation of the genome, this project was sparked by actually a capacity building project we led with a friend of mine, a Nigerian national who was working at ILRI. We developed a long training program for young or early career scientists in Africa, whereby we equipped them with skills in bioinformatics. Now, bioinformatics is the science of the automated treatment of biological information, and first and foremost, genomics information about genomes of species that you have to analyze to understand where are the genes providing susceptibility to a disease, where are the genes controlling the development, etc., etc. It is crucial that we are able, as Africans working in Africa, to do the necessary work on our species of interest, like indigenous species. In the world of crops, we can talk about coffee, of course, this LabLab bean. We're also working on African yam bean. And in the world of animals, we have animals of very high importance to the development of agriculture in the next years, because we have those animals that are more tolerant to heat, for instance, than their equivalents in Europe or in the US. Our cows here produce less than the cows in Europe or in the US, but they are more resistant to diseases that are around here, to the heat, and with climate change we'll have more and more heated areas on the continent. And also the African continent, harbors a huge biodiversity that is untapped. So if we want to find the next superfood, or if we want to find agricultural species of agricultural interest that will help us face the challenges coming ahead of us, mostly due to climate change, we have to study the biodiversity of species we have as indigenous species in Africa. And LabLab, the LabLab bean, is one of them. And we have also other projects on which we are working pertaining to other species.
Great. So I read in the article that there was some focus on identifying and modifying the genetic materials that contribute to the climate resilience of the LabLab in order to boost its nutritional content while maintaining drought resistance. If you could explain briefly in basic terms how you achieve that with genomics.
Sure. The genome is like a map. Researchers trying to understand how the biology of a species works, those researchers are blind if they don't have the genomic information. The genomic information means the genomes, where are the genes located on the different chromosomes, how are the chromosomes organized in terms of blocks of information, and those genes present on chromosomes, as I said earlier, they really are the cookbook for every single biological process taking place in the crop or in the species of interest. So, for instance, a species like lablab, like any other legume, actually has quite a high protein content. So we can study the pathways that contribute to the synthesis of those proteins that are then nutritionally extremely important. So in our study, we identified a group of genes called trypsin inhibitor genes that are responsible for the synthesis of an anti-nutritional factor. So there are some compounds, chemical compounds, that may be good for the plant against pathogens and insects, but are not so good in terms of the consumption of that plant by humans or by animals, because they reduce digestibility, they reduce nutrient availability. So typically it is very important that, thanks to our study, we unveiled the location of the genes responsible for the synthesis of these anti-nutritional factors, so that later on breeders can be more informed in their crosses when they select varieties of lablab in order to improve such aspect of the plant, digestibility, etc.
Thank you. So that's clear. So are there other objectives apart from like breeding and ensuring that you retain the best traits in crops and in animals? Are there any other objectives?
So genomics is key also to studies pertaining to the analysis of the biodiversity within a given species. All populations of a given species harbor some diversity to a certain extent. And in order to study this diversity, what scientists do is that they take reduced snapshots of the whole genome of a given cultivar, if we're talking about a plant, and they get snippets of the genome and they map these snippets to a reference genome. If they don't have a reference genome, they cannot map those snippets. So we provided a reference genome for the lablab gene that will enable scientists to characterize the diversity of lablab bean within East Africa, for instance, or within West Africa. And so we understand better what are the differences between the populations and within the populations we have, again, to inform breeding, but also to inform studies pertaining to heat tolerance. Lablab is quite heat tolerant. We have to study the genome now to understand what are the parts of the genome that help provide this high tolerance to high temperatures. And later on, those traits will even be transferable to species close to lablab, other legumes, for the purpose of fighting against, or adapting, more than fighting, adapting to climate change.
Great. Getting back to the point you raised about biodiversity, what is the expected long-term impact or global impact for countries in Africa having access to this information?
That's a great question. Currently, 3 crop species are responsible for providing the vast majority of calorie intake for human consumption. Maize, wheat, and rice are those key crops across the globe. Now, some of these crops require lots of watering, for instance. These are water-hungry crops. Lablab is quite able to sustain dry, long periods of drought, and so it's able to cope well and to develop even in an environment under high climatic stress. So it's important that in the next decades, at a global level, we diversify our sources of food, and perhaps some cereals, some legumes that are grown in Africa will become of an increasing importance to the African consumers, to the African markets. Currently, Africa is importing lots of those three main crops, maize, wheat, and rice. And we can be hopeful that by increasing the amount of research, increasing the amount of informed breeding on the continent, based on foods that are indigenous to Africa, will help countries in Africa reduce their dependency to major imports of crops coming from outside. So that's very important for Africa to be able to develop a more efficient path from farming to access to the market for key crops that are underused, a bit like forgotten crops, such as Lab. So if we increase the amount of research on Lab and other indigenous African crops, we can be hopeful that we will be able to reduce the dependence of African markets to imports from outside.
Yes, that's an excellent point. So thank you. So please describe the equipment you use just briefly and its suitability for the work being done, as well as the drawbacks. I think I saw that you said you used the nanopore.
Yes, so for this work, one key enabler for this work was the ability to sequence whole genomes thanks to portable and affordable equipment. So we use the Oxford Nanopore technology, and here in my hands I'm holding such a sequencer from this company, a sequencer called the MinION. We've been using long reads, so this sequencer enables us to read the genome in very long stretches. And that was key for us to be able to assemble the right picture of the full genome. Previous technologies were generating only short reads, so short fragments, short readings of the genome at a time. And those technologies relied, or do rely, because they're still quite pervasive, rely on large-scale, heavy, expensive devices. So in contrast, we really tapped into those handheld devices with cheap consumables that we were able to run in Africa.
Okay, and are there any drawbacks to using that technology? Because I understand it's not 100% accurate, so what are the drawbacks and what are the impacts if there are any?
So genomic sequencing is never an easy task when you try to sequence whole genomes of complex species. So each sequencing platform comes with its pros and cons. Oxford Nanopore technology generates long reads, but the accuracy of the readings is relatively smaller than accuracy of the readings given by previous technologies, what we call second generation sequencing technologies, embodied by the Illumina company. This is less true today because there are improvements in the accuracy of the Oxford Nanopore technology, but it was quite clearly at the time when we performed our sequencing experiments. Therefore, we not only used Oxford Nanopore reads, but we used these Oxford Nanopore reads mainly to have long scaffolds, long stretches of DNA, and we corrected the readings to fine-tune them. We corrected them with previously existing Illumina reads. So as in any scientific endeavor, we sit on the shoulders of giants and we use previously published knowledge. So in this case, we used previously existing reads obtained by a different research team, but we managed to assemble the whole genome in 11 chromosomes, what was not done before us. So using the sequencers to read pieces of DNA is only the first step towards constructing the full image of the whole genome. We have to use our high performance computing equipment. So we use large computers, and we are fortunate to run and maintain some of this infrastructure here on our campus at ILRI Nairobi. We use these huge computers to piece together the small portions of the genome that we've been able to read through the sequencer. We also correct sequences using a mix of different technologies. So then at the end of that journey, we reached a full picture of a genome assembled in 11 chromosomes. And after that, we had to also analyze the series of letters A, T, G, and T on these chromosomes in order to understand where the genes are, what are the areas where we have transposable elements. So there is a whole analysis of what are the constituents of the genome that has to be performed. On this genome is on around 420 million letters, and we had to analyze all these with our computers. We reused previously established genomic information, and we reused that, adding some other information from long reads in order to get the whole genome. So bioinformatics is a key enabler for African sciences. And of course, in Africa, within the continent, we don't always have the capacity to generate information, but if we train enough our skilled young scientists, we'll definitely have the capacity to analyze this wealth of information. And nowadays, in the world of science, I would say that the ability to analyze existing data sets is at least as important than the capacity to generate primary scientific data.
Great. So finally, did the pandemic's emphasis on genomic sequencing, for the coronavirus, increase the pool of researchers in Africa qualified to do this type of work, or at least better prepared for training in this area?
So absolutely, the pandemic was a great accelerator for the generation of genomic information on the African continent. The last cohort of bioinformatics training we organized it in 2019. So right after that, we saw a number of our trainees jump into national programs where they participated in the national efforts across Africa to sequence SARS-CoV-2 genomes. And also, SARS-CoV-2 has been a great springboard for these Oxford Nanopore Technologies sequencing devices that were used a lot throughout Africa, especially in Nigeria, Kenya, South Africa, to sequence SARS-CoV-2.
Thank you for listening to this podcast. This was IPS Caribbean correspondent Jewel Fraser speaking with Dr. Jean-Baptiste F. Fellner at the International Livestock Research Institute in Nairobi, Kenya.